Good afternoon, everyone. As Anthony said, my name is Declan Patton. I come from the Skin, Wounds and Trauma, the SWAT, no less, research centre in the RCSI. And we do a lot of wound care research. I'm just going to talk about a couple of diabetic foot ulcer projects, though, today. We also have a particular expertise in medical device research. I'm Deputy Chair of NREC MD, and I think Fidelma mentioned earlier on about us and Ireland being open for business to funders. The NRECs, I know you had someone here yesterday from NREC. The NRECs have done much to make Ireland open for business. So I'm going to talk about two studies, one of which is complete and one of which is ongoing, and Professor Shreenan, sitting over there, is involved in the second study that's ongoing at the minute. So the first study involved the measurement of subepidermal moisture in at-risk feet to help clinicians predict unseen diabetic foot ulceration. And the second study that's just kicked off is imaging diabetic foot ulcers using a deep view device which measures blood flow in and around an existing diabetic foot ulcer. So the DFU and subepidermal moisture, I'll just refer to subepidermal moisture as SEM, so the PI was Pauline Wilson on that one. So just a couple of background points. We wanted to look at what was the clinical utility of SEM, which is local tissue inflammation, measurement for the detection of foot ulcer development among adults with diabetes. We've done a lot of work in the pressure injury, pressure ulcer space, and we have found that elevated subepidermal moisture scores predict early stage tissue damage that is unseen to clinicians. And we sort of want to find out now if the same exists or can exist with diabetic foot ulcers. The SEM scanner itself is a handheld device, non-invasive. It measures early stage inflammation, and we have found that it can predict unseen damage up to 6 days before clinicians see it. That's in pressure injuries. And this is where we're ultimately going to go with diabetic foot ulcers. So there's the SEM scanner itself. It's quite small, actually. There it's a recharger, and it has you'll see the little circular piece at the top. That is the piece that touches the at-risk site on the foot. So the two clinical aims of this study: what was the profile of subepidermal moisture readings across a population of people with diabetes using the SEM scanner, and does using the scanner correlate with traditional methods of DFU risk assessment, so loss of protective sensation, pulse and temperature, for example. It was a prospective observational study. 216 patients kicked off the study. It took place in an acute hospital in Dublin, in the diabetic foot ulcer, in the diabetes clinic. All the patients that took part were classified as low, moderate, or high risk using the Scottish Risk Classification Scale, and patients who were moderate or high risk were followed up for a 7-day period with 2 further SEM measurements. And for those of you unfamiliar with the SEM scanner, which is probably everyone here, the SEM scanner will give a score ranging from 0 to 1, and anything over 0.5 is danger. And that's the score we used here, the cutoff point we used here as a high risk was 0.5. So we found that SEM readings across the patients of varying risk showed that the range of SEM scores differed right across those at risk, with low risk patients scoring an average of 0.26 and high risk patients scoring an average of 0.36. And you'll see the p-score there beside that, so there was a significance in that. There was agreement between SEM risk scores and traditional methods of assessment in over 60% of cases. And of the 13% of patients, 9 patients who developed a DFU within the 7-day period, all had an elevated SEM prior to that developing. Just in summary then, diabetic foot ulceration is a significant clinical problem that does increase morbidity and mortality. Prevention of diabetic foot ulceration can be missed in practice. The use of technology that can quantitatively detect an early stage biomarker, in this case subepidermal moisture, has the potential to radically reduce DFU numbers in at-risk patients. And whilst tentative, this study has shown that measuring SEM levels, local inflammation, in at-risk patients has the potential to be at least as good as traditional methods of assessment. So it's the first study. That study is done. So this study is just kicked off, and it's the wound assessment using spectral imaging. And the purpose of this study is to build a database for the development and validation of a machine learning algorithm for diabetic foot ulcers, healing assessment using images obtained from the DeepView device. So in the long term, the purpose of this study is to develop an algorithm that will tell clinicians what to do next. That's the long-term goal of this project. So right now, the early stages of the project is two endpoints. The primary endpoint is to assess the strength of the relationship between the DeepView device images and DEFU healing following 4 weeks of normal treatment. And that is measured using images and treatment records. The secondary endpoint is the exact same, only over a 12-week period, again using images and treatment records. And Professor Shreenan sitting over there is driving this project in his clinic. So the study design itself, now I didn't include an image of the DeepView device. It's like an ultrasound machine, but if you Google it, you'll see it. This study, just like the previous one, in fact, is fully funded and sponsored by industry, both US companies. And this study, the first study, took place during COVID, which was significant. And this study went through NREC-MD, which was also pretty significant. So the study design itself, so the DeepView device is indicated as an aid for the management of diabetic foot ulceration by providing information on wound healing potential within an image. It's not a standalone prediction device or anything like that, though. This is an observational study, and again, 144 patients with a diabetic foot ulcer will be enrolled over approximately a 1-year period. So in terms of measurements then, this study has just kicked off, so we can't say an awful lot other than what we intend to do. So at baseline, patients will be screened and consented. Eligible patients will have their wound imaged at baseline, and then further images will occur as the patient returns to the clinic in the subsequent 12 weeks. So care will continue as normal. There's no extra treatment, no extra visits to the clinic. Every time the patient comes to the clinic, they have their wound imaged. The study will end at week 12, or if the wound closes before that, and as I said, there's no impact at all on usual care. So the patients that are going to be included, people over 18 years, they have diabetes with a slow healing wound that is full thickness and at least 4 weeks old and is located on the plantar surface of the foot or toes. If 2 ulcers exist, well, then the largest ulcer will be the target one. The wound must currently be undergoing usual care and the patient is able to consent. And then in terms of exclusion, if patients will be excluded if any of these items exist. So if the wound is caused by a medical condition other than diabetes, if the wound is suspicious for cancer, if the wound has untreated osteomyelitis, and if the patient is pregnant or breastfeeding. Going back to Fidelma's interesting point earlier on, so that's all I have to say. I suppose just finally, we are in the space of predicting diabetic foot ulcers before they happen and predicting care pathways that clinicians can follow. So thanks very much for listening. Thank you.